Hello, my name is Biostella, and our concept is capsule networks, and that is what I'll be taking you through today. I'll explain capsule networks and how they can be used to detect diseases in plants like banana wilt. That's the project that we're doing. Um, let's go to the introduction. Capsule networks, or the CapsNet, can be shortened as that. And they were created by Geoffrey Hinton. They're designed to fix problems in the traditional CNNs, and that's the convolutional neural networks, the CNNs. The key idea of these capsule networks, the capsules understand both the presence of a feature and its details. Here, unlike the CNNs, the capsules, for them, they check if a feature is there. For example, the position, the angle. Yeah, they can analyze tiny disease patches on banana leaves while understanding the bigger picture. Then the reason as to why we prefer the capsules instead of the CNNs, because the CNNs have some limitations. One of them is that they lose special relationships between features. They can miss like the where of features, like where exactly in the leaf. Let's say you take a picture of the leaf, they don't know exactly where the disease is found in the part of that leaf. Unlike for the capsules, they can tell you the exact position where maybe the wilt has been discovered. Another thing is the max pooling in CNNs. They discard important details. Here, they discard the details during pooling. That's basically it. And they struggle with rotated or angled images. Because for the capsules, no matter what angle you put the image, it can still detect the diseases. But for the CNNs, they still have some issues with the angles and stuff. Then capsule networks keep all these details intact, making them more reliable than the CNNs. How this capsule networks, how it works. A capsule is basically, they're like a small team of neurons. Each capsule detects a feature. Yeah, each capsule detects a feature like a spot or a store, like a spot, and stores its details in an activity vector. Then they decide how their findings connect to bigger features, like a wilting pattern. Here are a small group of neurons that work together to detect features and their properties. Then the activity vector, a way to describe what a capsule detects, such as the shape or direction of a disease spot. We also have the dynamic routing. Capsules decide which higher level capsule to send the information to based on an agreement. Go to the next slide. And we're going to see how these capsules, they route information. Capsules communicate in a refined way. They send predictions to higher capsules. Predictions are weighted based on agreement with others. Then the process is repeated to refine connections. An example of this is a lesson. The capsule routes information to a disease. If if it agrees with other findings. When a capsule is detecting lesions, it sends information to a higher capsule that identifies the disease. How the capsules handle transformations. Capsules can identify features even if they appear in different ways. That is, if it's rotated, scaled, or moved, unlike the CNNs, as I explained before. An example is a healthy leaf looks flat and green. Then a diseased leaf has spots and wilts, no matter the angle of the photo. Capsules keep these relationships intact. The next slide. Then there's also something called the sparse capsule network. Capsule networks activate only the relevant capsules needed to analyze an image. These ones only focus on the capsules needed for a task. This makes them faster, more efficient, and easier to understand. For banana wilts, the sparse capsules highlight the key, the key diseased area. They don't highlight everything on the leaf. And detecting banana wilt with capsule networks. It also has its merits and demerits, or its problems and how that problem can be solved. Um, Identifying signs of banana wilt like spots. Capsule networks detect details like spots, discolorations, wilting, then they combine these into a pattern. This pattern helps identify banana wilts even in a low quality image or under different lighting. Then we have the capsule network, also has its architecture. The input layer, it prepares the plant image for analysis or processes plant images, resizes, normalizes. Then the second architecture is the primary capsules. They detect basic features, that's the edges and the spots. Then the disease capsules, they combine features to identify the wilt. Then the fourth one is the output layer. It decides if the plant is healthy or diseased based on all that. This is all. My colleague will take you through the other parts. Thank you.
Hey guys, so we are going to continue with our discussion of capsule networks. So we've already introduced capsule networks, and now we are going to look at the steps that are used in the implementation of capsule network model. So the first step is you obviously have to prepare your data. You have to collect images of healthy and diseased plants. So in this case, we are using our proposed machine learning projects to explain this concept. So we are going to be relating it to the banana leaf disease diagnosis project that you are doing for the machine learning project. So first we shall collect images of healthy and diseased plants. That's like a data set that contains images of healthy banana plants and also the plants that are diseased. Then we shall apply data augmentation This includes various rotations, cropping, and also adjusting the lighting of these images such that the model isn't frustrated while learning them. So the second step is we shall build the model. So we shall use capsules to detect features and relationships between the leaves and the leaf textures and everything. So then we shall train the model to identify bacterial wilt based on these features and relationships between the leaf patterns and textures and all. Then the third step is we shall deploy the model. Yes. So we shall deploy the model and optimize it to run on devices like mobile phones, which are more portable for farmers to carry around other than maybe laptops and tablets. Why choose capsule networks? So here we are looking at the advantages of using capsule networks as opposed to using traditional convolutional neural networks. So the thing with capsule networks is they are better at handling changes, which means they are robust to different variations. So they understand features no matter the position or orientation. So whether you change the lighting of an image or you change its dimensions, other capsules are still going to understand the features nonetheless. So the other advantage is that it gives explainable results, whereby you can see which features on the image, the features on the leaf images that led to the decision. For example, like it could have spots or discoloration or some kind of lesion patterns, which can show that yes, this is why the model decided that this leaf is actually diseased and not healthy. So the other thing with capsule networks is they are lightweight. They are lightweight models. Sparse capsule networks use fewer resources, making them suitable for use. So they use fewer resources because they use sparse capsules. Like they intentionally reduce the number of capsules in the network, and which increases computational efficiency, obviously. And then they also prevent misclassification due to overfitting. And the thing with sparse capsule networks is our model doesn't have to worry about overfitting because it solves all these problems for us. So even if your data set is a limited data set, the sparse capsule networks will definitely optimize the number of capsules in the network to prevent this misclassification problem that occurs due to overfitting. So the other advantage is that there's no loss of spatial relationships between leaf features like lesions and textures, unlike in traditional convolutional neural networks where there's loss of spatial relationships between these leaf features, and therefore the model is definitely going to misclassify these images. So how the whole process works, the capsule network model. So we assume that we've built a capsule network model and it has different layers. So if I'm to input a certain image, and if let's say the image is of a healthy leaf, so the model is going to activate only a few capsules. So this will show that this is a healthy leaf. The model will show that this is a healthy leaf. Then a lesion on the leaf will activate lesion capsules, and this will be a bit more. So this will show you that uh, this leaf has lesions, so it could potentially have a bacterial wilt. Then multiple lesions activate wilt capsules. So then the model concludes that this plant has bacterial wilt based on the fact that it has identified multiple lesions on the image of the leaf that you have sent in as input. So I'm going to give a brief example of how the capsule network model works, but it's not exactly our model. This is just a snippet of code to show how capsule networks actually work. So assuming this code is going to be used for our model. So first here we see that we have a capsule layer class. The capsule layer class, it's a single class that is responsible for learning high-level representations of disease symptoms such as yellowing, lesions, streaks on the leaf. So it's the capsule layer is going to learn high-level representations of these symptoms, and then the capsule layer also improves robustness to variations like lighting, like occlusions, um, like rotations. Then it's also responsible for capturing spatial hierarchies. I'm talking about this class here, the capsule layer class. It's responsible for capturing spatial hierarchies like lesion patterns, yellowing, streaking in leaves. So that's what the capsule layer does. Then we shall go to the second one, which is where we initialize our capsule layer. So this setup, it allows the model to dynamically learn important disease-related features, unlike traditional capsule networks, which depend on fixed hierarchies, and therefore it's not going to dynamically learn anything. It won't dynamically learn stuff that you've put in afterwards. So we have the number of capsules here that are in the network. Num capsules, it shows the number of output capsules, and the number of output capsules represent different bacterial symptoms like lesions or yellowing or maybe bacterial streaks. So this num capsules defined here as the number of output capsules. Then in dim is the size of the input feature vector. Then out dim is the size of the output capsule vector. And this is for disease feature representation. Yes, so it represents different features of the disease. Then numRoot, this shows the number of input features that contribute to each capsule in the network. Yes. Then self-weights. Self-weights is defined here, is a transformation matrix that projects inputs to high-level representations. So it's going to project the images that you've input into this model and transform them using a transformation matrix to high-level representations so that features can be easily identified by the model. So this is where also capsule voting takes place. No, actually, sorry, capsule voting takes place down here. Then we are going to move on to computing capsule transformations. So each capsule gets a transformed version of input features, and this is the weighted sum transformation. This is a weighted sum transformation that's used to capture complex relationships between features. So the significance of this is for the capsule network to group features to detect groups of features that are together. So it will detect that if these groups of features are together, then this leaf probably is infected with bacterial wilt. Yes, so it literally like you can say like modeling interactions between the features of these leaves to give a prediction. Then here we have the dynamic routing mechanism, which we talked about in the previous section when we were summarizing how these capsule networks work. So here we have the dynamic routing mechanism, which allows the model to selectively pass important disease features to higher capsule layers. So it iteratively refines predictions such that important disease patterns are not lost, like bacterial streaks, those streaks in the leaves, so that they aren't lost. So what it does is it iteratively refines the prediction such that it's the important disease-related features that are mostly focused on by the model. Yes. So then this is where the capsule voting happens. The capsule voting happens so that the features that are most relevant are the ones that are greatly considered by the model. Then here we have the squash function, which is used for, this is the squash function that it's used for nonlinear activation. So here, nonlinear activation has happened and it helps to distinguish between the different diseases. Let's say if a leaf has this kind of lesion pattern, then maybe it's bacterial wilt. Then if this leaf has maybe this kind of lesion pattern, or maybe there's some yellowing and everything, then this must be maybe fusarium wilt or segatoka. Yeah, so it distinguishes between different diseases of the bananas. Then it also produces normalized output here. This is where we normalize our outputs, so such that capsule vectors represent probabilities. So the significance of this is that it amplifies important disease features while it suppresses noise. So this noise is what could lead to misclassification, but that is what we are trying to prevent with our capsule network model. So it suppresses noise and prevents misclassification, which would potentially lead to overfitting. So here, it's only the strong disease features that are highly considered by the model, such that when it returns the output, it's able to make an accurate prediction. So going back to our presentation, the results of this code. So we haven't properly like written code for our project. But if we were to put like, let's assume we were to put the correct inputs here, what this code would return would be a batch of capsule vectors where each capsule vector represents a high-level feature, like a high-level feature of a diseased plant. For example, like yellowing, the streaking. So it would be the output. Maybe you've put in, like, you've given the model input of maybe an image of a diseased leaf. So the output this model would return would be the number of capsule vectors, a batch of capsule vectors, each representing the features in that particular image. And then you would maybe predict that it would predict that this leaf has bacterial wilt, or this plant has bacterial wilt, or this plant has sigatoka, and so on. So going back again to our presentation. So as we are concluding, we are just going to give a few remarks about capsule networks. So capsule networks offer a powerful way to detect diseases, and not just plant diseases, but also human diseases for probably like other models. They offer a powerful way to detect such diseases because they are better at detecting complex features of diseases. Now, in the context of bacterial wilt, I would say they are better at detecting complex features in leaves that are infected with banana wilt. It's going to properly detect that these lesions or this kind of yellowing on a leaf represents that this leaf has bacterial wilt or this leaf has this sort of disease. Then they are better at understanding images than traditional convolutional neural networks and are efficient enough for real-world use. So we've talked about sparse capsule networks, which improve computational efficiency because they use fewer resources by optimizing, by creating sparsity. They literally like reduce the number of networks in the model to make sure that it's more efficient and better at understanding features in these different images. Then with capsule networks, finally, we can improve farming outcomes and reduce crop losses, which is ultimately the goal of our proposed banana leaf diagnosis system. So I'm going to stop there. Thank you.